Hi everyone, welcome to today's section. Today we are going to discuss about the fluorescence resonance energy transfer. Myself, Sri Lakshmi, second MSc Biology from St. Mary's College, Thrissur. FRET, that means the fluorescence resonance energy transfer, is a physical phenomenon whereby the energy is created by fluorescence excitation of one molecule which is transferred to an adjacent molecule. It is a non-radiative process whereby the energy from an excited fluorescent molecule, that is from donor molecule, is transferred to a second non-excited fluorophore, that means acceptor molecule in its direct vicinity. FRET is used as a molecular roller due to its sensitivity in the range of 2 to 10 nanometer. The energy transfer result in donor quenching and that will lead to changes in the fluorescence intensity and the fluorescence lifetime of the two fluorophore. The donor is directly excited, whereas the direct excitation of the acceptor should be avoided. For the energy transfer to occur, it must satisfy five criteria. That are the emission spectra of one fluorophore must significantly overlap the excitation spectra of another fluorophore. The donor and acceptor must be aligned so that an acceptor dipole can be induced by the donor. The donor or acceptor separation can be no greater than 10 nanometer. The donor has a high quantum yield. And the final one is the donor is saturated by the acceptor. Assuming that these criteria have all been met, then the energy transfer will occur, and it is described by using the Foster equation. That is, Et is equal to R06 divided by R6 plus R06, where R is the donor acceptor separation distance and R0 is the donor acceptor separation distance at which the FRET efficiency is 50%. There are generally three commonly used approaches for measuring the energy transfer. First one is decreased donor lifetime, measured using the fluorescence lifetime imaging microscopy, which require an expensive secondary equipment. The second one is decrease in donor emission intensity, which is also known as a donor quenching, measured by acceptor photobleaching. The third one is increase in acceptor emission intensity, which is also known as the sensitized emission. There are some factors that can affect the fluorescence resonance energy transfer. The first one is brightness of fluorophore. If the donor and acceptor exhibit significantly different brightness level, dual fluorophore imaging may result in one channel that is saturated while the other is dominated by systemic noise, as most instruments can measure only a limited dynamic range. Therefore, it is best to use a donor and acceptor that are comparable or brightness. The second one is fluorophore alignment. False negative FRET signal can occur when the proteins are indeed physically interacting, but the fluorophores are not properly aligned and thus are more than 10 nanometer apart. Therefore, it is particularly important to ensure that the fluorophore tagged antibodies do not bind epitopes that are at the far end of each protein. Third one is spectral crosstalk. It refers to the cross excitation between spectrally overlapping fluorophores, which can affect the validity of the FRET signal. Instances of spectral crosstalk include when the acceptor is directly excited by the wavelength region chosen to excite the donor, or when the fluorescence emission from the donor leak into the detection channel for the acceptor emission fluorescence. As some degree of spectral overlap between the donor and acceptor is necessary for FRET, spectral crosstalk will inevitably occur for any FRET pair and must be addressed when FRET is measured. The last one is donor or acceptor stoichiometry. If the donor to acceptor stoichiometry is outside the range of 1 is to 10, the FRET signal may be masked by the background of fluorescent labels that are not undergoing FRET. As a result, it is critical to first understand the expression level of your interacting proteins prior to doing a FRET experiment using native tissue or cultured neutrons. Next, we have to discuss about the applications of fluorescence resonance energy transfer. First one is FRET in protein studies. It is a tool for identifying the distance relationship and supramolecular organization of cell surface molecule. It is used to learn the folding and unfolding of molecules. The protein partners can be co-localized by FRET. Next one is in biological membrane mapping. It can be used to study clustering of receptors and variation in conformations of membrane. Application in cell biology. It is used to study the protein-protein interactions in living cells. It is also used to study the metabolic and signaling pathways. It is used to study the DNA modification and also used to measure the distance between domains in a single protein and provide the information about protein conformation. FRET microscopy has the ability to capture the fluorescent signals from interaction of labeled molecule in single living or fixed cells. In this slide, we studied about the advantages of FRET. It is relatively cheap to implement. The measurements are obtained very rapidly. It is very good at measuring the changes in distance. In this slide, we studied about the disadvantages of FRET. It works only if the fluorophore are in correct orientation. The size of probes can introduce the problems. It gives no information about which probe move. The pH is sensitive. Free fluorophores can mask the energy transfer. And the final one is free pair labeling needs to be bright with the donor completely saturated by the acceptor. Thank you.